Let's just dive right in. Forget the sci-fi movie CGI for a second. Today, we're stripping all of that away and looking at the hard, clinical reality of the microscopic war raging inside the human cell. You're future clinicians, right? So you already know intimately just how vulnerable our cells can be, and you definitely know the limits of the natural immune response. But what if we could rewrite the rules of engagement? We're talking about moving way beyond traditional pharmacology and actively re-engineering the structural defenses of the human cell itself. Think about it like this. For literally millions of years, obligate intracellular pathogens have been absolute masters at exploiting host cell vulnerabilities. You guys know the mechanism of viral tropism inside and out. But what if we could just change the locks? What if we built a synthetic fortress so tough that it neutralizes the threat before binding even has a chance to occur? That's exactly what we're exploring today, pivoting away from our natural biological susceptibility and moving toward engineered synthetic intervention.
So here's our game plan for this explainer. We're going to move fast through the biomechanics of the threat, touching on viral pathophysiology before we dive into how exactly we're engineering synthetic defenses and destabilizing that viral envelope. After that, we'll ground the whole thing in actual clinical practice with lipid nanoparticles and nanodecoys before wrapping up with a look at the structural future of medicine.
All right, Section 1, Viral Pathophysiology, the biological breach. Now, to engineer a defense that actually works, we've got to precisely define the threat. And honestly, it helps to think of a virus not just as some alien invader, but essentially as a microscopic flash drive. It's loaded with malicious code, wrapped in a protective protein shell, and it relies completely on hijacking the host cell's transcription and translation machinery to run its payload.
If we break down the anatomy of the virion into its functional parts, it's pretty wild. You've got the lipid envelope acting as the outer protective barrier, housing that toxic viral RNA. But the real evolutionary masterclass in forced entry, that's the spike glycoprotein. These structures are perfectly designed for high-affinity binding to specific host receptors, like ACE2, and they trigger the exact conformational changes needed to force membrane fusion.
Once that jagged key turns the biological lock, the standard pathology is ruthlessly efficient. The spike glycoprotein targets the receptor, binds, and then the viral envelope fuses with the cell membrane. It injects its toxic RNA payload right into the cytoplasm. And just like that, the host cell is hijacked. It gets turned into a mindless factory pumping out millions of new variants, which inevitably ends in cellular lysis. The cell just bursts.
Which brings us to Section 2, Engineering Synthetic Defenses, Beyond the Immune System. Let's pivot from the biological problem to the synthetic solution. Pathogens have spent eons evolving to exploit our soft cellular targets. But what if we just upgrade our cellular armor using human-engineered nanotechnology? The contrast here is actually staggering. On one hand, you have the natural biological lock, like the ACE2 receptor. It's a soft target with a huge biochemical vulnerability to viral spike binding. On the other hand, imagine a synthetic wall. We're talking about an engineered polymer mesh made from lipid nanoparticles. It's basically an unbreakable geodesic dome. It doesn't just physically block the virus, it actively neutralizes the pathogen the second it makes contact.
Let's look at exactly how that active neutralization works in Section 3, Destabilizing the Viral Envelope, the Biomechanical Clash. Forget the cinematic explosions for a minute. We need to understand the precise biomechanics of what happens when a virion crashes into one of these highly charged synthetic polymer surfaces. It's an incredibly elegant sequence. First, the Nano-Shield detects the specific viral signature. Then, because the shield's surface is engineered with an active charge, it emits a localized electrostatic pulse. This pulse is brutal for the virus. It instantly destabilizes the lipid bilayer, essentially peeling the viral envelope right off. Without that structural protection, the viral RNA is totally exposed, and rapid enzymatic degradation by extracellular nucleuses turns it into harmless dust. The breach completely fails. Against this kind of engineered nanoshield, the ultimate biological assassin is reduced to absolutely nothing. The infectious potential is eradicated right at the cellular membrane before entry is even attempted.
Now let's pull this out of the theoretical realm. Section 4, Clinical Reality, Lipid Nanoparticles, Pharmacology in Practice. Because structural shielding at the nanoscale isn't science fiction, it's an active, foundational reality of modern medicine today.
Case in point, we already use lipid nanoparticles, or LNPs,
all the time in clinical settings. These microscopic lipid-based shields are engineered to wrap around fragile mRNA payloads. They pull double duty, protecting the mRNA strands from being shredded by extracellular RNases as they travel through the body, and making sure that payload safely escapes the endosome once inside the cell. So we are literally already deploying highly sophisticated synthetic armor at the nanoscale.
And that sets the stage for something even more mind-blowing. Section 5, Nanodecoys, the cellular sponge, trapping the virus. Here we're shifting from just protecting the host cell directly to deploying biomimetic competitive inhibitors that fundamentally redirect the viral attack itself.
Think of nanodecoys as literal flushable microscopic sponges. They are synthetic particles, perfectly engineered to flawlessly mimic the surface receptor profile of human cells, but they lack any intracellular replication machinery. They're totally empty inside. So the virions get tricked into locking onto these high-affinity decoys instead of your real vulnerable cells. Once trapped, these inert virus decoy complexes are just swept up by macrophages and harmlessly cleared out of the system. You eliminate the viral load without setting off some massive damaging inflammatory cascade. It's absolutely brilliant.
So what does this mean for you? Section 6, the structural future of medicine, a paradigm shift. As future physicians, you are entering an era of therapeutics where we simply won't have to rely entirely on chemical signaling or hoping the natural immune system can keep up.
The future of medicine isn't just chemical. It's structural. It's nanoscale. The wars fought inside our bodies are increasingly going to be won using highly engineered structural nanotech mechanisms. We are actively designing the physical architecture required to secure the cellular frontier.
So here's the question I want to leave you with today. If the future of medicine is nanoscale and structural, what microscopic battle will you engineer next? How are you going to integrate these advanced concepts into your own clinical protocols and patient care? It's something to think deeply about, because you are the generation that's actually going to deploy this tech. If you found this breakdown helpful for your clinical understanding, be sure to subscribe for more explainers on the hard science shaping the future of medicine. Stay curious, guys, and stay focused.